Hello all. In this video, we'll look into GATE Biotechnology 2021 paper, question number 17. The question is, the cellular process which utilizes RNA-induced silencing complex to block gene expression is option A, RNA editing, option B, RNA interference, option C, RNA polyadenylation, and option D, RNA splicing. So to understand that question, let's look into a mechanism called RNA silencing. So several terms are used to describe RNA silencing. There are three phenotypically different but mechanistically similar phenomena. So it's the coarse suppression or post-transcriptional gene silencing in plants, which is called PTGS, and in fungi it's termed as quelling, and in the animal kingdom it's termed as RNA interference. So let's look more in detail about RNA interference. So RNA interference or RNAi is a mechanism that inhibits gene expression at the stage of translation or by hindering the transcription of specific genes. So RNA interference targets, RNAi targets, they include RNA from viruses and transposons. So what happens here is the expression of a particular transcript is inhibited by the binding of several other molecules to that region. For example, if we are infected, the viral genomes get incorporated into our DNA. So our cell recognizes the foreign sequence, the foreign viral genome, and helps suppress the infection by this mechanism called RNA interference. So this is just one example of how it's important in the cell. So yeah, like we said, let's just look in depth of why this mechanism is important. So it's mostly used as a defense mechanism, like defense against infection by viruses. So it's used as a defense mechanism to protect against transposons and other insertional elements. And it's also used to control genome-wide regulation. So RNA interference plays a role in regulating development and genome maintenance. So 30% of the human genome is regulated. So we know that only less than 3% of the genome they code for proteins. So what does the rest of the 97% of the genome do? So they're all transcribed, and these are just non-coding elements, but they help in regulating these coding genes. So this mechanism of RNA interference was identified across a diverse range of species, ranging from fungi to insects to zebrafish and to higher organisms like mice. So there are several components involved in RNA interference. So we have certain RNA molecules. So these are the small interfering RNA or the siRNA. So these are double-stranded RNA molecules. There are 21 to 22 nucleotides in length. And we also have miRNAs or the microRNAs. So these are single-stranded RNAs that are 19 to 25 nucleotides long. So both of these are encoded by non-protein coding genome. So like I said, the mRNAs, they contribute to proteins, while the rest of the genome, the rest of the RNAs, they don't give rise to proteins, but they help in regulating the mRNA expression. We further have a protein called RISC. So this is the abbreviation for RNA induced silencing complex. So this complex cleaves the mRNA. And we also have certain enzymes called dicer and drosha. So the dicer produces a 20 to 21 nucleotide cleavage that initiates the RNA interference process. So we also have a protein called drosha. So this cleaves bases in the hairpin structure of the primary microRNA to form the precursor microRNA. So this precursor microRNA is later processed by the dicer protein. So these are the major components that are involved in RNA interference. Let us now look at the several steps involved in RNA interference. So in the first step, we have a double-stranded long RNA molecule. So this is processed into small interfering RNAs. So the small interfering RNAs are about 21 to 25 nucleotides in length. They have 2 to 3 nucleotides in the 3' end which are overhanging. So we could see that like in one of the strands the RNA is longer, but in the second strand it is shorter. So these are called 3' overhangs. So this conversion is done by the protein called dicer. So this is an RNase type 3 enzyme. So RNAs are molecules that degrade RNA molecules. So the next step is where these small interfering RNAs are associated. They bind with the RISC complex and they unwind. They form like separate sense and antisense strand. So the RISC complex binds to the small interfering RNA, and this double-stranded RNA unwinds and it forms a single stranded. And now the RISC complex is activated. The RNA-induced silencing complex is activated. It just has one of the strands from the small interfering RNA. The third step in RNA interference is where the antisense small interfering RNAs acts as guides for RISC to associate with complementary single-stranded mRNAs. So in the second step, we saw that the activated RISC complex consists of the RISC protein, which is formed with the antisense strand, one of the strands from the small interfering RNA. So this is the activated RISC protein. So this binds with a single-stranded mRNA, which is complementary to the sequence of the small interfering RNA. So what happens in the final step is that this complex it cuts the mRNA approximately in the middle region paired with the small interfering RNA. So the mRNA is degraded further. So this leaves the mRNA transcript non-functional. So this does not give rise to any protein, and hence the RNAs interfere, and this RNA is silenced. So it doesn't produce any proteins further, or it is just non-functional. So this is the whole mechanism of RNA interference. So there are several therapeutic applications of this RNA interference technology. They could be used in various fields like in hematology, in oncology, that is cancer research, in stem cell biology, and in infectious diseases. So a lot of hematological disorders result from loss of gene function or mutant genes or degraded or absent genes. So the RNA may be used to create models. The RNA interference technology can be used to create models of disease processes and could help develop pharmacologic and genetic therapeutic targets. So in oncology, this technology could be used to target oncogenes. So the cancer causing genes could be targeted and degraded and thereby could help prevent the metastasis or further progression of cancer. So it could also be used in stem cell research and can be used to treat infectious diseases like targeting certain viruses. In this video, we looked at the process called RNA silencing and the components involved in it. So coming back to our question, the cellular process which utilizes RNA-induced silencing complex to block gene expression is, so RNA-induced silencing complex is RISC, and the mechanism that uses this complex to block gene expression is RNA interference. So this answers our question. So the other options, RNA editing, RNA polymerization, and RNA splicing, are other post-transcriptional modifications that happens to the RNA transcript. So thank you.